Hello and welcome to this lecture. Today we are going to discuss inflammatory bowel disease. We will start with that. What is inflammatory bowel disease and how can we differentiate between its two types? One is ulcerative colitis and the second one is Crohn disease. We will describe the epidemiology of IBD. How can we describe the pathogenesis of the IBD and the morphological and clinical characteristics? So it defined as a chronic condition resulting from inappropriate mucosal immune activation, and it has been classified into either Crohn disease or ulcerative colitis. We will discuss the case study in our upcoming lecture, especially on ulcerative colitis. So the first thing we need to know is that it is the inappropriate immune activation, and it has been classified into two types. One is Crohn disease and the second one is ulcerative colitis. And if I take a look on the Crohn disease and ulcerative colitis, you see the main difference from the look is Crohn disease is in the form of patches. You can see these in the intestine in different patches, whereas ulcerative colitis is actually in the form of sequential. It is continuous, sequential, continuous, and continuous involvement till in the beginning in the rectum. Then we can see that the inflammation will be transmural, ulceration will be found. And why we mentioned transmural, it means that it can go in different mural surface with ulceration fissures, like you can see this finger-like projection again. So the main thing, if you see these finger-like projection like this, so they are showing you the presence of the fissure. Then because that fissure is not restricted to one membrane, it is restricted to the different others membrane or mural surface. So we mentioned transmural inflammation. So this is, you can see this ulceration formation and pseudo polyps formation. So if we see the difference between chronic disease and ulcerative colitis, the main difference, the region. Crohn disease occur in the ileum and colon. It is actually, I already mentioned, if you see the picture, it is segmented. It means it occur in different parts. Whereas ulcerative colitis is only in colon. So that's the main difference. If you take a look of picture, ulcerative colitis is only in the colon, whereas Crohn disease is not restricted to colon, it restricts to ileum as well. What is meant by ileum? It means that it also occurs in the small intestine, and ileum is the one section of the small intestine. Then the rectal involvement in Crohn disease is sometime, whereas ulcerative colitis is always. As if you take a look of the picture, we mentioned there the beginning in the rectum. So again, that ulcerative colitis involves the rectum, whereas in Crohn disease, it's sometime yes, sometime no. That's why we mentioned this skip lesion. Why we mentioned skip lesion? So it could be possible in the rectum, sometime it could not be. That's why we use the word skip. And as we know, it is an English terminology. You can skip the sound, skip the music. So skip means that you can skip this section. I'm not going to discuss all of them. I'm going to discuss only major perspectives. The one is the bowel region, that is very important to know. Then the rectal involvement is important. Distribution, I already mentioned that skip lesions, and ulcerative colitis is more diffusive. Then the bowel appearance, that is the next important thing, is thick, whereas in ulcerative colitis it is thin. The inflammation, it is very important, is transmural. I already mentioned it. What is transmural? And I already put the definition below, you can see. Whereas ulcerative colitis is limited to mucosa and submucosa. Pseudopolyps formation is moderate in Crohn disease, whereas it is marked in ulcerative colitis. If you take a look of these both pictures, this left picture is actually showing you the presence of transmural inflammation, Whereas in the right side, the picture showing you of the ulcerative colitis has the formation of these pseudopolyps, and they are more restrict to mucosa and submucosa, whereas Crohn disease is not restrict to mucosa and submucosa. It can go deep into transmural and the other sections of the tissues. So you can see that this dark red structure is showing you different sections of the tissues, and it can penetrate deep to the other mural surface, the other tissue sections. Whereas if you take a look of ulcerative colitis, it is only restrict to mucosa and submucosa. Then when we talk about the ulcers, they are more deep knife-like. I already mentioned finger-like projection like this, very sharp, as it is transmural. So obviously it go more deep. So that's why they look like a knife-like structure. Whereas ulcerative colitis is more superficial because it is mural, and you can see that submucosal. So that's why they look like a formation of pseudopolyps. Then there are some other inflammatory mediators. Lymphoid reaction is marked in colon, whereas it is moderate in Crohn disease, whereas it is moderate in ulcerative colitis. Then fibrosis is marked in Crohn disease and moderate in ulcerative colitis. So if you compare even the features, you easily said that the Crohn disease is more severe as compared to ulcerative colitis. So this is more severe if you see the other characteristics. Then we see some other characteristics like fibrosis, granuloma, serositis. You can take a look. It is marked in Crohn disease as compared to ulcerative colitis. So this is the pictures how the formation of pseudopolyps will be there, because this is example of ulcerative colitis, which is superficial. And inflammatory bowel condition actually is the root cause of toxic megacolon, as we know that because it is a medical condition which causes swelling and irritation in section of the digestive tract and can be extremely painful and permanently damage the small and large intestine. Epidemiology, more common in females. I'm just going to read through. It is occur in young adults, more occur in whites, 3 to 5 times, and more often in European ex-colonized Jews. More prevalent in North America, Northern Europe, Australia. And what is the reason behind IBD? It's the hygiene hypothesis. What is hygiene hypothesis? We discussed last time that with this picture you can take a look. It's very easy that people are not exposing to the environmental microorganisms, because nowadays people are more careful of their hygiene. But what happened that at adolescence or at young age, because people are more protective, more careful with the microorganism, so they are not exposed to them. So that is why if they are not exposed to the microorganisms, their body, their immune system have not been developed. So when suddenly a microorganism affect, because you are eating the food, microorganism can go inside through your food. So what happened then, that it can trigger then immune response, abnormal immune response. So that is why the scientists suggest that people need to, or kids or young people, they need to expose with the allergens. That's why this picture is so funny, that they're talking about, can too much hygiene create allergy? So he said doctor prescription. So because he's have inflammatory bowel disease. So you can see that this is a new hypothesis, which is known as hygiene hypothesis. But again, in the current scenario of COVID-19, currently this scenario, if you cannot expose to microorganism now, why? Because Crohn, this disease, COVID-19 is more dangerous and deadly. If you expose to microorganisms like this, maybe you fall with more severe illness. So that is why it is very, very important. The pathogenesis mechanism is the errant host interaction with intestinal microbiota and intestinal epithelial dysfunction and abnormal or aberrant mucosal immune response. So when we take a look of the pathogenesis, we need to understand in detail that how this happens. That, for example, the bacteria are present on the mucosal surface. They enter inside, and the barrier, so they lead to inside, uh, and then they activated macrophages and tumor necrotic factors, and then they activate macrophages first, and with the initiation of the macrophages and the bacterial components that goes inside, they react with the dendritic cells, and they form the complex with the T cells, which we know that in which we know this is used as adaptive immunity. So these microbacteria, they are responsible to form the complex with the T cells at CD4 with the help of CD4 cells, and then this T cell is further responsible for the production of interleukotriene 12 and interleukotriene 23 that promote the production of further abnormal immune response by producing Th17, Th2, and Th1. And Th17 is responsible with the help of interleukotriene 17 to produce neutrophils, and it will produce in two ways. One way, either with indirect mechanism through the formation of the binding of the dendritic cell with the T cells to produce neutrophils, or it can be produced through direct interleukotriene 8 that is responsible to produce neutrophils. Then you can see that the Th2 cells with the help of interleukotriene 13, it go back, and with Th2 and Th1, they are responsible for the production of tumor necrotic factors and interferon gamma to produce more macrophages. So you can see this is a cyclic mechanism. And I have talked very complex thing in this slide. I just make it simple for you. The simplest thing that you need to do is, first you need to see that there will be production of immune response, and I'm deleting my highlighting, and I'm going to revise again so that you understand that in simple terms how you understand this mechanism. In stage 1, we know that the bacteria are present on the mucosal surface. They enter, they cross the barrier, and when they cross the barrier, what is the thing that the bacterial components, they activate the attachment of the dendritic cells with the T cells, and this is they binding with the receptors, and then they make a complex. This is the first thing you need to see. The second thing, you have to understand that they are responsible with the help of interleukotriene 8 to form neutrophils, which is another inflammatory mediators. And the process does not stop there. Process continues with the help of binding with the T cells through CD4 receptors. They are responsible to produce further Th cells, which is actually, you can see Th17, Th2, and Th1. But you can see that when the T cell bind with the dendritic cells, it also activates interleukotriene 23 that promote further Th4 cells. So you can see that the production of Th17 cells, and if you take a look that the process does not stop here. Interleukotriene 23 that is produced at the same time when the dendritic cells are binding with the T cells, and interleukotriene 23 they bind with the Th17 cells, and then they are responsible for with the help of interleukotriene 17 to produce more neutrophil. So this process, by producing neutrophil through IL-8 to IL-23 to indirectly with the help of T cell to produce more neutrophil, is cyclic. So that's one cycle that is going on. On the other side, if you see on the right hand side, they are forming with Th1 cells with the help of interleukotriene 12, and then interferon gamma is responsible to produce more macrophages and then more tumor necrotic factors. This is another cycle on the extreme right side. And in between the cycle, you can see that cycle that is promoting the formation of Th2 cells. It work with the help of interleukotriene 13, and it go back to the mucosal surface. So it means that we having three cycles going on. Cycle number center, where the formation of the dendritic cells to T cells formation complex, that is not normally going on. Second side, on the left side, is the formation of neutrophil, which is on the extreme left. Then on the middle side, there will be two intermediate mechanisms with the help of Th17 and Th1 that work with numerous ways. Like Th17 is responsible for producing more neutrophils. Then intraleukotriene 12 with the help of interferon gamma producing more macrophages. So again, they are responsible for producing more immune mediators. And the Th2 is responsible with the help of interleukotriene 13 to go back and affect the mucosal surface. And they may be responsible for making more permeability for the microorganisms to enter. So again, this is a very complex mechanism, but you need to understand very basics that how this happens. The basic thing is that the formation of more neutrophils and the formation of macrophages. And the third thing that is actually, it is going in a sequence. Everything is going in a sequence, that the dendritic is the dendritic cell and T cell complex. So that's how that mechanism occurs. Morphology, the common site is usually affected in the terminal ileum, ileocecal valve, or cecum. So use a picture, physiological, or this is the anatomical picture, that this is the this is the ileocecal valve. It look like a feet-like structure, crow feet-like structure. So we mentioned crow feet folds. Then it occurs in the terminal ileums. This is a picture we already discussed before. How gross structure look like? It look like small intestinal stitcher. It's just like that somebody has stitched the surface. Linear mucosal ulcer will be formed, and thick intestinal wall. As you see, the walls are quite thick. And we already discussed the characteristics. If we go back, in Crohn disease, the bowel appearance will be thick, but in ulcerative colitis, it is not thick. It is thin. So you have to understand that we are discussing a journal morphological characteristics. So they are talking about linear mucosal ulcers, thick intestinal wall, then creepy fat, these lumps. They are showing you the fat-like appearance and greasy appearance. The another thing, they are showing you the presence of cobblestone. Again, what is cobblestone? Cobblestone is another artifact. It is a metaphor. It's just like a stone-like appears, and if you see through endoscopy, through biopsy, they found like same structure like cobblestone. That is why we discuss this in terms of cobblestone. When we discuss the histopathology, it looked like the cells, Paneth cell metaplasia, cluster of neutrophils within a crypt referred as crypt abscess, and then we have non-cheesiating granuloma formation. So the main thing is that it is a metaplasia of the Paneth cells, formation of the crypt abscess because of the presence of the neutrophils. As if we take a look on our pathogenesis, we already discussed the excessive production of neutrophils. This section is important. So there will be neutrophil production because through two mechanisms via interleukotriene 8, are either by making complex with the dendritic cells of the T cells and promote the neutrophil production. So this is very important, and they are expressed in the form of crypt formation or cleft abscess formation. And then we are also showing you that there will be non-cheese-eating granuloma. It means that it is not cheeseous in nature, not cheese-like granuloma formation. The clinical features consist of the intermittent attack relative to the mild diarrhea, fever, abdominal pain, the lower quadrant pain, especially fever and bloody diarrhea, 20% will be found. The disease reactivation occur because of the stress, such as physical or emotional stress, or dietary items, or smoking if somebody have. And in case of iron deficiency anemia, there will be the decrease of the albumin in the blood, so we can express as hypoalbuminemia. We discuss about the nutrients malabsorption or malabsorption of vitamin B12. So if, for example, someone they have iron anemia or hypoalbuminemia or malabsorption of the nutrients, and that result in deficiency of vitamin B12 and bile salt, then it could be IBD will be occur. So thank you so much. In the next lecture, we will discuss its subclassification. Especially, we are going to discuss ulcerative colitis, and I will bring with one case study. So thank you so much.